Welcome. Thank you for being here. I'm Gemma Thompson. We're going to spend the next 6 minutes on the physiological mechanisms and effects of transdermal direct current stimulation on chronic pain. Let's jump in. tDCS uses a montage of electrodes placed on corresponding treatment areas of the scalp and directs a current no higher than 2 milliamps into the corresponding treatment area in the cortex. For pain, these are usually anodal stimulation on the C3 and C4, or M1 for fibromyalgia. M1 and M2 are the mastoid processes. These are the bony prominences behind the ears and are also referred to as A1 and A2. The current either decreases or increases communication between neurons. For an action potential to initiate, the input must depolarize the cell enough to reach the threshold of excitation. And as part of the all or none law, not every input is strong enough to tip this threshold to release the action potential. In a maladaptive network, the signals that we want to initiate might not be strong enough to reach that threshold. So instead of sending an action potential, the signal dies in the cell without sending its intended communication. If you can imagine the cell is like a cup that must overflow to send its signal. Anodal tDCS fills that cup in the neuron just enough so the biological signals that would otherwise not be strong enough can make it through to their intended destination. tDCS also works in the opposite direction. Sometimes problems are caused by the wrong signals making it through a gateway. Cathodal tDCS artificially empties the cup, inhibiting the inputs from reaching the threshold, creating an artificial IPSP. When thinking about the role of anodal versus cathodal stimulation, imagine you're standing at the edge of a lush, dense tropical jungle. Anodal stimulation takes a tiny track and allows the brain to build a boardwalk. Cathodal stimulation artificially ceases communications between neurons, as well as reducing blood flow, allowing the strong path to crumble and the jungle to regrow. We know that the networks in our brains change in response to stimuli in a process known as neuroplasticity. When it comes to chronic pain, we're working with maladaptive plasticity. That means the brain changes as a response to prolonged physical, social, or emotional stresses, and the connections made within the network during this time become unhelpful because neurons that fire together wire together. The pain associated with a condition can persist even after the event has resolved. This maladaptive plasticity can lead to conditions like central sensitivity, fibromyalgia, even phantom limb syndrome. The most common treatment for pain in about 70% of cases is a prescription for opiates or other pharmaceuticals, like gabapentin or even tricyclic antidepressants. These interventions have significant side effects and, in many cases, manage symptoms, but do not treat the underlying cause of pain that may stem from maladaptive plasticity. At this point, it's important to mention that chronic pain is complex, and the issue is not always in the nervous system. For example, a diet high in amines, glutamates, and salicylates, common chemical compounds found in fresh, healthy food, can trigger unexplained pain events in sensitive people without triggering the immune system. Although the treatment is exciting, we cannot group pain under a single cause and effect umbrella, which is important to remember when selecting subjects for trials and interpreting results, especially for complex conditions like fibromyalgia. However, tDCS could be helpful diagnostically when attempting to rule in or out the underlying causes of complex conditions like fibromyalgia. If a patient responds well to the relatively quick, cheap, and painless intervention, we could design treatment modalities on the assumption that the issue lies predominantly in the maladaptive nervous system rather than another underlying cause. tDCS has been proposed as a therapeutic tool for pain syndromes. Although initial results in human subjects are encouraging, it still remains unclear whether the effects of tDCS can reverse maladaptive plasticity associated with chronic pain. A team at Harvard have investigated whether tDCS can reverse chronic stress in the pain system using rats to investigate behavioral corticosteroid, interleukin-1 beta, and tumor necrosis factor alpha, using a 4-way controlled rat model of chronic restraint stress. In the study, the stress groups were subjected to 1 hour of restraint 5 days a week for 11 weeks. We don't know how rats conceptualize time, but if we look at the biological timescale differences between rats and humans, each hour for the rat might be the same as a person being in a constant state of stress every hour of every day for 5.5 days. Following 20 minutes of active treatment per day over 8 days, the rats showed a significant decrease in hippocampal tumor necrosis factor and behavioral changes, becoming consistent with the control group, supporting the notion that tDCS can reverse the maladaptive plasticity associated with chronic stress. However, the duration of this change was not observed. It's important to note that this is an animal model, and the 20-minute treatment time in biological timescale would be closer to 9 hours of treatment for a human. However, this is not replicable because after 26 minutes of a nodal treatment and 20 of cathodal, the neurons exhibit an opposite response. We don't know why this flip occurs. It could be an interesting area for further study, but may be difficult to obtain ethics approval. The analgesic effect of tDCS could be assisted by attuned effects in pain sensation in multiple neurotransmitter systems. Overall, the findings of this case study show a positive effect of tDCS on pain, although the mechanisms underlying this effect remain unclear. The evidence suggests that they include non-synaptic and synaptic mechanisms alike. On top of the neuroplastic changes we discussed earlier, the non-synaptic mechanisms would include changes in local ionic concentrations, which could arise from changes in the transmembrane proteins and exposure to a constant electric field, as well as increased blood flow.